Hello, everyone. Welcome to our interviews for the South American Mycorrhizal Research Network. Today, I have the pleasure to be with Bala Chaudhary, who is Associate Professor in Environmental Studies in Dartmouth College in the US. Welcome, Bala. Thank you for being with us today.
Thank you for inviting me.
Bala, you recently published a very interesting review called Fungal Dispersal Across Spatial Scales in Annual Reviews. And I wanted to know first why you decided to publish a review on fungal dispersal.
So research in my lab has been focusing on dispersal now for a few years. And so my students and I had just been thinking about dispersal all the time, and dispersal really as a understudied mechanism of structure community assembly for fungi, and in particular what my group focuses on, which is mycorrhizal fungi, and we focus on arbuscular mycorrhizal fungi, or AM fungi. And so we had just been having lots of conversations about dispersal, thinking about different methods, thinking about spatial scale, and it just seemed like a good time when we were synthesizing a lot of literature and reading a lot. It seemed like a good time to put out a review paper on the topic.
And so when people think about fungal dispersal, of course, they think about spores. So can you tell us a little bit about what are the different mechanisms that fungal use for dispersal?
Yeah, so certainly people have been studying fungal dispersal for a long time. But the point we make in the paper is that macrofungi and fungi that form above ground fruiting bodies or specialized structures for dispersal are comparatively studied more than other fungi that are below ground fungi or cryptic fungi that still are kind of important in ecosystems, but also are relatively understudied, and we don't know much about their dispersal mechanisms. And so, yeah, I would say that a lot of the fungal dispersal ecology work does focus on spores, but one point we make in the paper is that, of course, the spore is just one part of the fungal body, and that we really need to be thinking about other parts of the fungal body, like the mycelium, or in the case of mycorrhizae, colonized root components or plant parts that are colonized by fungi. And so those are all potential propagules. And so if you think about really the definition of dispersal, which is movement of an individual or a propagule, then we should be thinking holistically about fungal propagules.
And so for fungi, of course, individual species or individuals move around, but then you also have the case when you have entire communities that are going to move. So do you think this is also a very important component of fungal dispersal, that a lot of species actually need the entire community to be moving?
I do, and I think then it helps us think about not just fungal communities moving, but fungi moving with bacteria, or microarthropods moving with fungi and bacteria, and how those communities may depend on each other and really need to move together. And not only do I think that that's an important part of fungal dispersal research, I think that's a way that really general dispersal ecological paradigms are changing. And that this idea really coming from the microbial world has the potential to think about how we understand dispersal of all life on Earth.
Mm-hmm. And so now if someone listening to this interview who wants to go deeper into fungal dispersal, what are the fundamentals that people should start looking at if, for example, they are working on a certain ecosystem or certain species and they want to know more about fungal dispersal?
Excellent question. And that was actually another major goal of the paper, to really try to stimulate ideas and stimulate thought in this area, because I think it's a great line of research. And so I would say to somebody like that, get the paper and take a look, because not only do we present a lot of research questions that are underexplored, but also we present some frontiers in the research that are new directions of ways that people are studying fungal dispersal, and also new methodological advances. And so I think it's actually a very exciting time to be studying fungal dispersal, because we have the emergence of multiple different technological advances that are kind of overlapping and coming together. So one of those is, well, of course, much more accessible and affordable environmental genetics techniques. And then also kind of a growing trait-based analytical framework. We also have these large and growing kind of global environmental sampling networks that can be leveraged to study movement and dispersal of fungi. And then lots of novel environments that are being explored for fungal dispersal, like the atmosphere, like oceans. And then I think there's also cool new research on studying fungal movement at very small scales using microfluidics techniques. And then I think also just the way we think about systems in terms of social ecological systems. So what are really the causes of fungal dispersal? How are humans moving fungi, and how does fungal dispersal affect human health and other social systems too? So there's a lot going on, and lots of places where students with various interests can choose different entrance points to the field.
Yeah, you also showed in the review that there is a little bit of regional bias, right? With a lot of studies coming from the US, some studies from Europe, from Australia, but then there's also entire regions that are still very unexplored. So do you think it is possible that there are like specificities between regions, and if someone wants to study fungal dispersal in other places, he may not, or she may not find the same result as, let's say, northern temperate region, for example?
Yeah, I mean, 100%. And so, we did this bibliometric analysis in the paper where we looked at what some of the general kind of themes in fungal dispersal research were, also where most of the research is happening. And like you said, there are a lot of gaps on who is doing the work, or I should say, whose work was captured in our search. Of course, there are biases in the way we searched, 100%. But yeah, I have no reason to believe that we wouldn't find different patterns. So for example, in some of the previous work in my lab, we found that soil disturbance can be potentially an important way that soil fungi are released into the air or liberated to facilitate long distance dispersal. And that may not be the case in other locations, or maybe this dispersal mechanism differs. And so I think, studying these phenomenon in many different locations is very important.
Mm-hmm. Yeah, and then finally, I have kind of a personal questions that I have been wondering for a long time. So I would like to know your opinion about it. There is some kind of paradigm in mushroom, for example, saying that most spores are falling very close to the sporocarps, and then very rarely you have events of long distance dispersal. So I'm pretty sure that different groups of fungi probably have different strategies. But I was wondering if I can have your opinion about, do you think that long distance dispersal is rare in general in fungi?
Yeah, so I think, and I hope that we tried to dissect this a little bit in the paper. I hope that came across that, there are multiple mechanisms of long distance dispersal, right? So there's wind dispersal, which could be long distance. There is water dispersal that can be through freshwater systems. It can be through ocean currents. There's also animal vectors, so birds could potentially be long distance dispersers. And one thing we tried to do in the paper was present how spatial scale, like you say, is very important to thinking about dispersal and what the most important vectors are, or most important mechanisms of dispersal are. And I guess I would say what we put forward has very little data to support it.
Yeah.
And so, there's this general idea in dispersal ecology that abiotic mechanisms drive long distance dispersal and biotic mechanisms drive shorter distance dispersal. But I don't know, I think that it's still poorly tested with fungi. And then, of course, I think it's also really difficult to generalize broadly about fungi. I was just invited to a recent working group on dispersal, and I was asked to represent all of fungi.
Yeah.
But there were people there, like there was a fish person, there was a butterfly person, and I was like, listen, you all work on animals. You wouldn't ask this person to inform on another person, and I'm being asked to represent all 3 to 13 million species of fungi. And so I think sometimes that can be frustrating because we'll say, oh, the idiosyncrasies prevent us from speaking about generalized patterns. But I mean, it is what it is. We're talking about a huge group of organisms.
Yeah, exactly. So at the end, to go back to the paradigm that you mentioned at the beginning of your annual review, do you think that everything is everywhere but the environmental filter, like the theory that we have about microorganisms?
Right. So Boss-Becking hypothesis. We think about applying Boss-Becking hypothesis to fungi because that's sort of the dominant paradigm for microbes. But what we say in the paper is, are fungi microbes? Like sometimes they can be microscopic. Spores are microscopic. And sometimes a fungal individual itself could be microscopic, a hyphal network that's maybe 1 meter squared as a leaf endophyte. But then on the other hand, the largest terrestrial organism is a fungus. And we know that individuals can be quite large. And so it doesn't make sense to me that the Boss-Becking hypothesis, which is really based on unitary, discrete microbial individuals, would necessarily apply to fungi. And sometimes I wish this is going to get me in trouble, but sometimes I wish the Boss-Becking hypothesis would just die. But it is a nice kind of thought exercise. I wouldn't call it a testable hypothesis because we certainly can't sample everywhere and look for every species everywhere. But it's a useful thought exercise, particularly when we're thinking about what are the controls of community assembly for fungal organisms.
Yeah. That was very appealing. Thank you so much, Bala, for your time. And we will be back soon with more interviews for the South American Mycorrhizal Research Network. Thank you, Bala. See you.
You're welcome. Thanks for asking.